Protein synthesis, translation. Inside the body, the process of translation occurs within every single cell. Each cell has a nucleus. After transcription, the mRNA is moved out of the nucleus through nuclear pores and enters into the cytoplasm. This mRNA strand now acts as a template for protein synthesis. An enzyme called amino-acyl tRNA synthetase is present in the cytoplasm. This enzyme has two binding sites. One of the sites recognizes the amino acid methionine. This is followed by binding with ATP molecule and release of pyrophosphate. Finally, the tRNA and the activated amino acid bind it together. This amino-acylated tRNA is called met-tRNA and releases from the enzyme. This follows the commencement of first stage of protein synthesis, that is the initiation stage. During the initiation stage, a smaller subunit of ribosome binds to the mRNA strand. This mRNA strand is made up of codons. Again, the codons are made up of 3 nitrogen bases. Then the ribosomal subunit moves along the mRNA in 5' to 3' direction until it recognizes the start codon or AUG codon. At this point, the methionine tRNA possessing anticodon UAC pairs up with the AUG codon on mRNA. Then the larger subunit of ribosome binds with a smaller subunit. The larger subunit of ribosome shows 3 sites: acceptor site or A site, peptidyl site or P site, exit site or E site. This whole unit forms the initiation complex. This is followed by elongation stage. During this stage, another tRNA molecule carrying amino acid approaches the mRNA ribosomal complex and fits into the A site. Then the bond is formed between methionine and tRNA containing amino acid. As a result, methionine tRNA deacylated. The deacylated tRNA now moves to the E site, from where it dissociates. Meanwhile, another tRNA carrying amino acid attaches to the A site. This is followed by binding of peptide bond with the amino acids. This process leads to the formation of amino acid chain. This event is called elongation. Finally, when the UAG codon or the stop codon reaches the A site, elongation process is terminated. Termination is the last stage of protein synthesis. The chain of amino acid molecules releases from the ribosomes. The released amino acid chain forms the protein through modifications. This part of protein synthesis is the translation. Then the tRNA detaches from the mRNA, ribosome detaches and dissociates into smaller and larger subunits.